In this webinar, you're going to hear from Dr. Jennifer Bone and Dr. Michael Lotze from UPMC. They'll be presenting data on their study using a natural language processing model for early prediction of patient responses to IL-2 immunotherapy based on motif elucidation of CDR3 TCR and BCR clonotypes.
Okay, why don't you go to slide 2, Jen? And so I just would like to thank the group shown here who've been working for the last few years on trying to identify aspects of what we call the adaptome, which I'll introduce a little bit later, and how the T cell and B cell repertoire changed during IL-2 therapy. Jen Bone, you're going to hear from extensively. She was a graduate student when most of this work is being done. She's now actively working at Nurix, as I am, which is a small molecule drug discovery company. Her mentor during her PhD thesis was Newell Washburn, who's shown to her right. I'm there in the center for some reason. I'm a professor of surgery, immunology, and bioengineering at the University of Pittsburgh, but again, right now as Chief Cell Therapy Officer at Nurix. None of the presentation today will actually have anything to do with Nurix Therapeutics. The real hero working behind the scenes is Miranda Steele. She's the one who organizes everything at iRepertoire and keeps the team moving all the way through this last pandemic that we've been working through effectively, and was able to make sure that we obtain data from our patients treated with high-dose IL-2. And then Pranav Murthy, who was working on his master's during the period this work was done, who's also now working as a scientist one at Nurix Therapeutics. Next slide. So let's introduce this idea of the adaptome. Everybody's familiar now with the genome and perhaps the microbiome and other omes. And so we invented a term to try and define what is the total aggregate of functionally diverse B cells and T cells, which have rearranged T cell receptors. The great gift of evolutionary biology, going back now 500 million years ago to jawless fish, was the idea that we could generate a very diverse repertoire of T cells and B cells. And if you look at fundamentally what happens in an individual, essentially everybody has essentially a unique adaptome. Everybody generates in a random way their rearranged T cell receptors and B cell receptors, which are in turn shaped by additional factors, and that includes the Manhattan of the genome, which is the greatest diversity in all of our genomes, which are the major histocompatibility loci on the short arm of chromosome 6. That's class 1 and class 2 molecules. It's something you may be familiar with. The individual antigenic exposure history, as well as exposure of an individual's mother, actually. And then the constant fine-tuning and regulation that occurs of the immune system. But what's going on underneath the hood is this random VDJC recombination between genetic elements that allows you to generate as many as 10 to the 15th to 10 to the 25th different T cell receptors and B cell receptors. And if you look at any two individuals in the population, they share at most 3% of their individual T cell receptors and B cell receptors on average. Identical twins that are genomically identical only share 6% of their T cell receptors and B cell receptors. Now, interestingly, you start off with an extraordinarily diverse set of T cell receptors shown in the gray graph in the bottom right. And as you age, you refine and narrow your diversity based on an expansion of individual T cell and B cell receptors that recognize antigens from your environment. So everybody coming through the latest pandemic will know that many of us are now exposed either naturally or through the vaccine to have specific T cell receptors and B cell receptors recognized. And just like cardiac function decreases during age, kidney function decreases with age, lung function decreases with age, your natural immunity and the adaptive immune response also decreases with age, as shown nicely in this figure. Next slide. So let me introduce the notion of high-dose IL-2 therapy. This is something near and dear to my heart since we started this therapy when I was at the surgery branch of the National Cancer Institute back now, I hate to say it, 30, 40 years ago. And it was approved in 1993 in patients with kidney cancer who had disease that was no longer able to be surgically resected, and then a few years later in patients with melanoma. And it had been, up until the era of checkpoint therapy, a mainstay for treatment of both of these tumor types. Unfortunately, as a high-dose regimen, IL-2 is associated with significant toxicity, substantial toxicity that occurs in the lung, the brain, the heart, the liver, the kidneys. Virtually every organ is affected transiently. And we believe that that's associated with this cytokine storm that changes the biology of each of these individual organs. And although there have been multiple attempts over the last couple of decades to try and limit that toxicity, none of them have really been effective in terms of reducing side effects without affecting the anti-tumor effects. And so our current notion, and they're good mouse models that we've published on, is that this IL-2 treatment is associated with what we call a systemic autophagic syndrome. And for those familiar with apoptosis, which is programmed cell death, autophagy is programmed cell survival. Essentially, cytokines inform each of the individual cells within organs to shut down normal function and to go into a period of programmed cell survival. When you stop the IL-2, virtually all of these changes reverse. And quite strikingly, this is the first patient I treated with high-dose IL-2 alone. This young man had widespread pulmonary metastatic disease and a very similar looking liver that looked like Swiss cheese. And with a single treatment over just a few weeks, all of his cancer went away. And you can see this is a picture 2 years later showing all of his disease has gone. Really remarkable. And said, here was something for the first time ever to really see these extraordinary responses, but only in these diseases, melanoma and kidney cancer for the most part. Next slide. So just to introduce the history of IL-2 therapy, here you see a whale breaching the Golden Gate Bridge. It was a picture done by John Wehrle back in 1980. It served as sat in the boardroom of the CDS Corporation, CDS meaning whale. Their original plan as a recombinant DNA company was to clean up oil slicks using recombinant enzymes. Can you click once, Jen? And so our first IL-2 therapies were done with a Manhattan-like project with IL-2 that was made from the Jurkat cell line by about 30 scientists over 6 months at DuPont that we gave to patients in 1983. Next, click. Tada Taniguchi, working at the University of Tokyo, cloned IL-2 that same year. Next, click. Roche in-licensed Tada Taniguchi's patents for recombinant IL-2. Next, click. CDS developed a pegylated IL-2 that was originally thought to be a winner for therapy. And in actual fact, Roche decided that they would trade for Cedus's new invention, which is called polymerase chain reaction, developed by a Nobel laureate at Cedus, for the rights, granting Cedus the rights to IL-2. Next click. Chiron acquired Cedus back in the middle 90s. Next click. Novartis acquired Chiron, as is often happens with big pharma, and then out-licensed it, next click, to Prometheus. And then worldwide rights have recently been acquired after Nestle acquired Prometheus by a company called Clinigen. Next slide. So again, to make a point, since Jen is going to be talking about our results in kidney cancer patients, here's one of those dramatic responses. What you see here is a CT scan on the left showing a huge tumor in the liver. That big black amoeboid structure is a metastatic kidney cancer. And here you can see IL-2 therapy has allowed complete response of that tumor. And I don't know whether you would call it a cure or not, but that patient is still disease-free more than 20 years later. Steve Rosenberg recently wrote a single-author paper summarizing advances in another disease, melanoma, showing how little response there was with melanoma vaccines back in the pre-80s. The response to IL-2 that allowed 20 to 25% response rates. The advent of checkpoints, including CTLA-4 and PD-1 antibodies, followed by the emergence of adoptive cell therapy, which Jen and I are both working on now, tumor-infiltrating lymphocytes alone, along with non-myeloablative chemotherapy. It's really been a remarkable history in terms of how these therapies have evolved. Next slide. This is a recent study showing that even in the period in which kidney cancer can be treated with multiple other agents, including checkpoints and what are called TKIs, tyrosine kinase inhibitors, that we still see a full 25% at least response rate, with both CRs, shown in the top line, and PRs, with substantial responses blended in that green line in the middle. They're of substantial duration. These are not just days or weeks. These are months of survival. And you can imagine this is a very effective therapy, which is now associated with keeping the field alive, waiting for the checkpoints and adoptive cell therapy. Next slide. So why IL-2, why adaptome, why kidney cancer? Essentially, high-dose IL-2, which is both a T-cell growth factor and a B-cell growth factor, will promote the expansion of those cells, what we call the adaptome. And this is a limiting factor in the tumor microenvironment and can rescue patients by expanding those cells, enabling them to be fully functional. In addition to promoting the proliferation of both CD4 and CD8 T cells, it also can promote the expansion of NK cells, which we believe are associated with much of the toxicity associated with IL-2 administration. Although you do see Treg cells, which are the gluttons of immunobiology, they eat the IL-2, they appear to play a modest role in melanoma and probably a less important role in patients with kidney cancer. Next slide. So just to reiterate, the guts running underneath the hood of a T-cell response is shown here on the right with an alpha and beta T-cell receptor recognizing peptide antigen presented in the context of an MHC molecule, and that's both for CD4s and CD8s. It's part of a complex that we call collectively the CD3 molecule, which consists of a number of other chains, including the epsilon chain, which is the target for many of our CD3 antibodies. And essentially, the great wisdom of evolutionary biology in the adaptome is it allows you, without ever having been exposed before over your evolutionary history to a virus or a bacteria, to respond within days to promote the acquisition of an effective immune response. And the main question that Jen and her colleagues tried to address is whether a deep dive into the adaptome might be a strategy to understand why some patients respond to this therapy and some do not.
Sure. Thank you so much for the introduction. So as Dr. Lotze said, we're looking for, how IL-2 treatment is functioning in these patients and why certain patients are responding and not responding. So basically in this work, we are really searching for these biomarkers related to the adaptome for predicting the patient response to IL-2. And to test this theory, we started with a cohort of 29 patients with progressive renal cancer, and we administered high-dose IL-2 treatment with the autophagy inhibitor hydroxychloroquine, as you can see on the right here. And we ended up with a final cohort of 20 responders and 9 non-responders to this treatment. The patients were assessed at least 365 days after treatment, in which their tumor size was measured and they were classified. So the question becomes, how do we measure the adaptome? What we ended up doing is we took PBMC samples, just intravenous blood samples from these patients at what we call day -14, which is our pre-treatment time point, day 1, which is the first day of IL-2 treatment, and then day 15, which is 15 days post our first IL-2 treatment. And what we then ended up doing is basically a next-gen sequencing along with this DAM-PCR, which I'll describe in a moment, to elucidate the CDR3 regions that we think are the variable regions responsible for carrying the information related to cancer neoantigens. So to start, we looked at day 15 and asked the question, can we, just based on the adaptome, the T cell and B cell receptors from the adaptome alone, predict and classify these patients according to the response that we recorded 365 days later. So just a little more about the CDR3 region. So this is the region of our T cell and B cell receptors that we think are most responsible for carrying the information of different cancer antigens. And as Dr. Lotze eloquently described earlier, these are what we these are made from VDJ recombination and a couple of other different types of variable insertions and deletions to create this humongous plethora of different T cell and B cell receptors. And we can make up to 10 to the 25 different B cell and T cell receptors in our bodies, throughout our adulthood. So the question becomes, how do we quantify the adaptome? And this goes back to the next-gen sequencing and DAM-PCR methods that we talked about earlier. And this is something that Miranda has helped with and that iRepertoire has been really integral in helping us to elucidate. So we start with our frozen PBMCs, and we essentially are trying to extract the T cell and B cell receptors from our alpha beta chains from our T cells, our gamma deltas from our T cells, and then our heavy, light, and kappa chains from our B cells. And what the DAM-PCR really allows us to do is essentially remove PCR noise from our product and really help us narrow down, which CDR3s are coming from which which CDR3s are real, essentially. And we are able to do this by essentially having a unique molecular identifier that gets mapped back onto our genetic information that helps us to really narrow down our CDR3 content and then also the clonotype number. So what do we get when we get this data is we get our CDR3 is represented here as a sequence of amino acids, and the subsequent read counts, basically the number of times that the CDR3 is observed in the sample. And here you can see that they're ranked in terms of how clonal, how often they appear. And if I were to take all of these CDR3s from this one patient sample and try to visualize them, we get what we call these tree maps that in which we have all these little circular rectangles are a different CDR3, and then the read count is related to the size of the rectangle. So here you can see this patient has a pretty diverse repertoire. And this is all their T cells and B cell receptors combined together. And you can see that there really isn't any, too many large rectangles here that demonstrate that there's, kind of a selection for a specific CDR3. But perhaps that might be different in patients that are battling some type of cancer like renal cancer. So how do we quantify this information? And what the current assessment methods are to take these diverse tree maps and or the actual clonotype numbers and frequency itself and collapse it down into a single number. So 3 of the current metrics that we have been using for looking at the diversity of these samples is D50, diversity index, and entropy. And what these metrics allow us to do is to look at the distribution of these clonotypes in a sample. And our hypothesis is that, if a patient is undergoing kind of an attack on the immune system, that we're going to see higher clonality or higher number of reads of selected CDR3s compared to, say, a normal patient that is not, battling anything at all. And so D50 allows us to look at the head of that distribution, meaning, really focusing on the clonotypes that are most frequent. The diversity index allows us to kind of look at the whole distribution, and entropy with this log 2 allows us to look at more of the tail of the distribution. But what we found is that actually the diversity metrics were not really able to help us classify these patients at day 15. And even if we were to see some significance in that they, so here we have our D50 and our entropy. And this is on the left for our T cell receptors, and on the right for our B cell receptors. Even if we were to have some significance values here, which we we did see for maybe some day -14 data points, it's really difficult to classify data that's not is not separable. So if there's any overlap between these red and blue responders and non-responders, our classifier gets confused. And so the idea is that we want to be able to separate this data in such a way that a computer can go in and say, patients with this score are going to be non-responders and patients with this score are going to be responders. So what we're going to have to do is transform this data into a space that allows us to separate it in a robust way. So there are some challenges for doing this. And, machine learning is near and dear to my heart. It's what I did my PhD on in a biomedical space. And there are some limitations to it that I've come across in cancer. And some of those limitations involve the idea that, as we increase our model complexity, we are also decreasing our feasibility of being able to throw kind of conventional machine learning methods at this problem. And the reason for that is actually related to the structure of the data. So if we consider all of our CDR3 sequences as equally likely to be immunogenic, then, when we have a very diverse sample, we have, up to 10 to the 25 different features in our matrix, and only say in our renal cancer experiment, 29 patients. So we get these kind of long and fat arrays that are kind of inappropriate for conventional machine learning. And so what I'm going to talk about is how we're going to overcome that problem, and then how we could maybe flip the data to use that to our advantage. So to start, I'm going to talk about these long and fat data sets where each CDR3 is considered a feature. And we're looking at an N of 29 patients and starting just with day 15 classification. So working with the repertoire, a couple more challenges to overcome, in addition to limited patients, is the idea that we don't detect every CDR3 every time we are looking at a patient. So I mean, even if you were to take intravenous blood from your left arm and from your right arm, you might not, just because of the low frequency of some clones, you might not get the same answer every time. And so not only do we have this very large and kind of fat feature space, but we also have different numbers of features for every patient. And this can be really challenging also in structurally for data science. So to address these challenges, as I said, we're going to map our data into our high dimensional space data into an intermediate space that's going to help us make the data more separable and allow us to classify the patients at day 15. And we'll also talk about flipping the data in a way that allows us to monitor a patient against themselves over treatment. We're going to narrow down our features in our first analysis with a carefully supervised selection that I will describe. And then we're going to show whether or not we were able to classify the patients correctly. So what is the problem with analyzing our diversity the way, our D50 the way it currently stands? And not that it's necessarily a problem, but we want to get more information. We want to squeeze as much information out of this data as we possibly can. So one way I thought that we might do this is to ask the question, well, what about what happens within a CDR3 sequence? Say we have two CDR3 sequences that are incredibly similar, but differ by just one or two amino acids. Is it possible that they are able to bind to the same HLA? And is it possible that, what I've been calling our cancer-specific message is actually embedded in these CDR3 sequences? And so actually what we would have, instead of a single immunogenic CDR3 sequence of amino acids, we would actually have groups of convergent CDR3s. And by convergence, I don't mean evolutionary convergence. Actually, this is an article that was published out of the Mark Davis lab back in 2017, in which they were able to correlate, have a structural basis for looking at the similar specificities of these groups of CDR3s. So what this means is that there's the potential that we can detect groups of CDR3s that could be immunogenic, that could have information about our patient response to IL-2 treatment. And that really allows us to narrow down our feature space. So when I talk about looking at inside the CDR3 sequence, I'm really talking about how do we find, these messages. So here on the left is an example of a kind of a made-up CDR3 sequence that has a made-up part of it that we would want to detect. And what we can do, and part of the natural language processing that that this talk is about, is being able to look at this text kind of in the same way that our word processors look at text. And this is by breaking them up into sections. And so here on the bottom, so this is a first order Markov model where, if we look at the full sequence, our k equals 0, this is, kind of what our diversity metrics are based off of. But then we can start to look at, well, what happens, if I have, just, one amino acid based on its previous neighbor? What about two amino acids based on its previous neighbor? And we can kind of get a sense of, the importance of a chain of amino acids in this sequence. And so actually this model that I developed was inspired by this exact method for detecting CpG islands, which is basically the search for high GC content using nucleotides. So this would just be ACGT. So we have a 4x4 matrix here. And the idea is that by looking at these kind of nearest neighbors of our sequence and mapping these and counting them in a specific DNA sequence, we can start to predict, what parts of our DNA would have high GC content and which parts would not. So here on the left is an example of a transition probability matrix for a CpG island, and you can see that in the heat map there's a high CG content, and then in the right here is outside of the island, and you can see a striking difference in less CG content. So my question became, could we do this for amino acids? And so instead of having a 4x4 transition probability matrix for ACGT, what if we had a 20x20 transition probability matrix that looked at all of our 20 amino acids? And so this is an example of a patient from this study and what their transition probability matrix looks like for their CDR3s for day 15 with all of the amino acids. So this is a 20x20 matrix now. And you can see that there are some light colored boxes here. And the question becomes, is this a pattern? Can we detect more patterns? And so what the probability, transition probability maps also let us do is kind of keep track of what types of patterns we have. So in this pattern, we have two what our previous analysis would call unique sequences, but actually you and I can see that there's just one insertion here. And so this comes out in the numerically in these matrices, as you can see on the left and the right. The first sequence has 0.33 and 0.66, and the second has 0.25 and 0.75. So this is a way for us to really have a lot of precision in differences in these sequences. Again, what if we have, the same sequence, but it's just a little bit jumbled? How does that change our analysis? And you can see here that, we have the same amino acids, but they are spatially located differently in these matrices, and that allows us, again, to have precision in how we look at the within sequence content. So here's an example of how the classifier is going to work for a responder and a non-responder for some kind of dummy data that I threw in here. And what I want you to notice is that the this kind of highlighted region here is visible in our non-responder and not visible in our responder as a way to help us see how these patterns are going to come out of this analysis. So what we did is we essentially did the K=2. We looked at every amino acid and with respect to its previous neighbor. So what that means is that we had a feature space of up to 10, for each chain, up to 10 to the 5 features. And we basically took each amino acid sequence and chopped it up into pairs 2 and put it into this transition probability matrix, much like I showed you before. We were able to do that for every single patient. So after we, so over here on the left is an example patient, and over here on this, starting up on the right column, is an example of 2 different patients and what their transition probability matrices look like. And interestingly, we don't really see any patterns by eye, but one great thing about computers, and one thing I love about these kinds of processing techniques, is maybe there's a pattern we can't see. And so in this next graph below is basically a filter that we created that looks for significance in patterns in these matrices. And what it pulls out is these pairs of amino acids that it predicts are going to be indicative of response or non-response. And so in the bottom here, you see a feature significance matrix that is basically a black and white version of these colored ones that only retain the significant features. And what we're going to do is overlay that matrix, that feature significant matrix, over our raw patient data, so that what shines through are only the patterns that are indicative of response and non-response. So when we do that, what we found is actually that, so here are 2 test patients, and don't worry too much about the numbers, but it basically just means our transition probability matrix for a delta gene at day 15. And we put them both through the same filter, and what's coming out here visually is specific patterns. You can see these bright yellow spots, and And for the non-responder, you can see that for the responder, you can see that the patterns don't really come out. And so what we can do is we can quantify this for all of the patients. And when we do that, we now have that separable data that I was really hoping for in the past. So not only did we find significant motifs that significantly separated the patients, we were able to do it for all 7 chains, actually, in this analysis when the chains were looked at independently. And what was really exciting was that each chain had at least 90% accuracy in being able to predict the patient response to IL-2 by day 15. So in conclusion from this first analysis, we had at least 90% accuracy in response classification for each chain. And what this really is interesting, it was kind of surprising because we thought, maybe only the T cells would be indicative of response, or maybe only, and so, it was interesting that the B cells were also able to perform pretty well. And so, we've been having some theories back and forth about, what that really means. Is there, is the adaptome orchestrating together? Is there co-stimulation of B cells with activated effector T cells, or is there T cell independent B cell activation? And these are questions, that we hope to be able to inquire on further. So in the limited time that I have left, I want to talk a little bit about a way we tried to cleverly leverage the data. So instead of having our fat short matrix that I showed before, perhaps what we could do is take a single patient and monitor them against themselves. And this essentially takes our matrix that was kind of difficult to work with before in terms of throwing machine learning methods at it and flipping it on its side. And so now we have this kind of like tall array that we're kind of used to seeing, and that makes our analysis a little bit more conventional. So we started with the same analysis again. We basically chopped up our CDR3 sequences into our amino acid pairs and put them into the same matrices. But now we're going to do it for all 3 time points and compare each time point to the previous time point. So now we're looking at day -14, day 1, and day 15 and asking the question, how does a single patient's adaptome change over time? What is the information content? How does it change over time? So here is a single patient with a transition probability matrix for each time point. And what this is trying to show you is that we compared, our day 1 to our day -14, and then we compared our day 15 to our day -14 and asked the question, are there any interesting patterns? What is happening here? And so to quantify this information, I'm going to tell you a little bit about this graph. So on the right, this is an example of an analysis for a single patient in which we compared their day 1 to the day -14 in this upper left, and our day 15 to the day -14 in the bottom right in the green. And what this top right corner of this graph is just an overlay of these 2 distributions on top of each other. And what you can see is this drastic change in the content of the CDR3 sequences, as you can see them overlaid on each other. And so we wanted a way to quantify that. So what we did is we basically plotted them against each other here in the scatter plot on the bottom left and asked, well, if they're exactly the same, if these distributions were exactly the same, we would have a straight line. We would have, we'd be able to run a Pearson correlation between the 2 and see a rho value or a strength of the correlation that would be really similar to 1. Everything that's, as we deviate from 1, essentially, as we get closer to 0, we see more and more of this kind of like circular splattering of data. And so this is how we tried to just kind of take a first pass at being able to analyze this. So here I wanted to show you some raw data of a single patient. So this is 7 chains. You'll count 7 matrices of these 4, of the one that I showed you before, of these 4 panels, 1 for each chain. And you can really see how the chains are changing. So, if we look over here at the delta chain, you see this kind of, again, paint splattering, same thing for the alpha chain, same thing for the beta chain, maybe a little bit closer together, same thing for the gamma chain. But when we look at the B cells, they start to really, with the exception of the this one here, like they start to really form this kind of expected, line essentially. And, we can visually validate that by looking at this upper right-hand corner here, where when I overlay the distributions, they're very similar. And so on the bottom right here is essentially what we collectively recorded all of these values for the kind of linearity of these distributions. And what you can see, what we would expect is that, if we had some kind of bias in the T cells or the B cells, that, we would see only a change in the B cells, only a change in the T cells, or only a change in the B cells. So in this patient, when we take all of those numbers here and we plot them, we see that actually we have correlation coefficients that are very low for just the T cells and very high for just the B cells. And we did this for all of the patients. And we found that this BCR and TCR bias, so I showed you an example of a TCR bias, but the reverse is also true, where we could have, this kind of step function going the other direction, where that would be high T cell, low B cell. And we found actually a significant difference, that this was actually a real effect that we were observing, and that this bias is something that we didn't expect. So there were 3 different states that we found: a BCR bias, a TCR bias, like I just showed you, and then a mixed bias. So there were patients that didn't have that difference in T cell and B cell response to IL-2 at all. So when we put all the data together for all of the patients, we found that only the responders had a clear TCR and BCR bias, and that only the non-responders, and that all the non-responders had a mixed TCR-BCR bias. And there were a couple of patients who did end up responding that had a mixed bias. And the question becomes, is this related to response? And we actually have a validation cohort that's coming in that we're going to be able to analyze. We're really excited to look at this a little bit further. But what I really wanted to point out is that, and what was so exciting is just this idea that the adaptome is this orchestration between B cells and T cells, and the data really reflects that. And as somebody who came into the cancer immunotherapy space kind of cold about a year and a half ago, it was really motivating and interesting to find this data, to find these observations, I should say. So in conclusion, we found distinct and significant bias in the TCR versus BCR populations in patients who responded to IL-2. We have a couple of theories about why this might be happening. We might be visualizing T cell recruitment to the tumor site, and that's why we aren't able to see changes in the information content in the T cells and the PBMCs. The validation of the role of this bias will be studied. The one way we could study it is by comparing clonotypes from peripheral blood to those found near the tumor tissue, and there are a couple of data sets we're trying to get our hands on for that. And again, just to reiterate, all 7 TCR and BCR chains were able to distinguish responders from non-responders by day 15. This was a really incredible result. And so again, we're really excited to validate this study in a future cohort because it suggests, again, an orchestral role of the adaptome between B cells and T cells. Future directions of this work I've touched on already. We are also looking at doing this modeling for the day -14 time point to see if we have any CDR3 predictors of patient response to IL-2, and this would be really incredible because we could potentially use that to screen patients before treatment. And then finally, as I'll say for like the 3rd time, we're excited to run this model on our validation cohort and really show our observations and the testing of our hypotheses on that.
So first question, is there any correlation of the repertoire modeling with blood samples in a 3-year follow-up for natural killer cells, MDSC, regulatory T, and dendritic cells in prediction of response?
I think that would be a really great study for us to run. We currently don't have enough data from the patients that we did have to run that study, but I do think that, you know, in the future, when we're starting to look for, you know, even further validation of this model, that we would absolutely want to run a study in which we had a multi-year follow-up, even just to see if there's a persistence of these CDR3s in, you know, in later time points after treatment.
Great, thank you. Do you have a feel for the relative contribution of sequencing errors to the plethora of similar but distinct CDR3 sequences?
Yeah, so to answer that question, I would first want to ask what type of sequencing errors we're referring to here, because, you know, one thing we really thought about, especially with working with iRepertoire and working, you know, with this data, is PCR error. So, you know, I think it really depends on if you're referring to that or if you're referring to, you know, kind of random recombination events that somehow, you know, make it into the bloodstream that maybe make it into our analysis in some way.
Okay, we'll see if a follow-up comes in on that for what they were specifically after. In the meantime, did you also try to use the GLI-pH motifs for classification prior to developing your method?
Actually, no. So it was interesting. So they're referring to the glyph analysis that Mark Davis did, which we're also very interested in. We've actually spoken to him personally a couple of times and are running, you know, some data through the glyph analysis currently. They actually have a glyph 3 now that's coming out, which we're really excited about. But again, I think, you know, I think it's another information study that's worth doing. It's kind of a low-hanging fruit for us, and you know, kind of to be coming. Preview to be seen in the next, hopefully, couple of months.
Great. So to follow up on that question about errors, they were specifically asking about PCR errors. So can you comment on how you use DAM-PCR to address those?
Yeah, and if I, you know, butcher some of the biology, perhaps Miranda or Dr. Lotze can jump in. But essentially, DAM-PCR is a way that iRepertoire came up with to help reduce PCR error by assigning a unique molecular identifier to to our amplicons, essentially. And those amplicons are then quantified by their unique molecular identifier, which helps us to essentially remove PCR noise and really be able to count on those kind of low frequency clones as real.
Excellent. As far as the specific amino acids that are contributing to these findings, you know, for each chain, is it mainly the contacting regions of the CDR3 or the IG, or is there, you know, maybe some more structural information that we could get about the chains that would augment your approach?
I think that's a great question. And this is a very naive approach. And I do think that when we start to put in structural information, even if we just weight, you know, the, you know, like the, you know, amino acid, like say like the 8th or 9th amino acid in, or, you know, the ones that we think are really going to be in contact, you know, I think we might even find a richer analysis. So I'm excited to embark on that. And I think the glyph analysis will help us with that too, because they're also looking at that.
Can you talk us through the rationale of including hydroxychloroquine as part of the IL-2 treatment regimen?
Yeah, so and again, you know, Dr. Lotze can jump in if needed, but essentially, IL-2 is fairly toxic. It kind of encourages this what we call autophagic syndrome that you know, is very hard for the patients to endure. And so what the hydroxychloroquine does is it's an autophagy inhibitor. And so by putting it with the IL-2 in treatment, we were able to give a higher dose IL-2 for a longer period of time, kind of more safe or with less toxicity compared to if we didn't.
And maybe I can jump in here, Jen. That was a perfect answer. That many of the toxicity, but also the efficacy of IL-2 therapy is limited. In part, this was all confirmed in animal models before we went into patients. But autophagy is a mechanism also for the tumors to avoid T cell and NK cell recognition and destruction. And so as a consequence, when you do autophagy inhibition, you enhance the ability of T and NK cell effector function so that tumors are more susceptible once they get activated with IL-2 and able to kill through granzyme perforin and TNF family members.
Next question is, have you attempted to correlate your data with biopsy analysis? Oh, you're muted, Jen.
You're muted, Jen.
Sorry. In short, yes, we were kind of wondering whether or not, those patients with the mixed response had, some change, in their tumor size compared, or how they were progressing compared to the rest of the patients. But we didn't have, I think, we will have to go back and see if we have the data to do further analysis. As of now, we have not done that yet.
Okay, kind of a broader question because some of the audience may not be super familiar with the term adaptome. Can you define that for us again and explain how it differs from a repertoire and why it's important to investigate?
Yeah, absolutely. So the adaptome is sort of a term that we invented. Not, and by we, I mean mostly Dr. Lotze and our, our team. But we use it to describe collectively the T cell and B cell receptors in our repertoire. So that is specifically what we're interested in the adaptome, and Dr. Lotze can jump in if I'm missing anything. But so when we say the response, we say a response with via the adaptome, we're really referring to in the study all 7 T cell and B cell chains.
And let me jump in for a minute. So many people who do repertoire analysis will only focus on a single chain, the beta chain of the T cell receptor, which is plenty diverse. It's a lot of information for people to keep track of. But as Jen mentioned, you have a much more diverse repertoire than just a single chain of one paired receptor. So by analyzing the alpha chain, the beta chain, the gamma chain, the delta chain, the heavy chain, the kappa chain, and the lambda chain, you really get a much better picture, and you learn things that you couldn't have learned otherwise. So for example, one of the bias of either a T cell response or a B cell response, you would never have known about had you not done a full 7-chain adaptome analysis. And I think our hope and expectation, especially after Jen completes her validation set that we are now embarking on analyzing, is to convince people that if you really want to understand how an immune response to a virus, to a tumor, to a transplanted organ is occurring, you need to evaluate all 7 chains, get a complete picture of the immune response.
And when you guys did this, did you notice, were there common clones for particular chains between patients that did or didn't respond?
So we did look into this, but we didn't, our sharing of clones is very low between individuals. And that's partly why we were motivated to look within the amino acid sequences of our CDR3s as a way to be able to compare similar clones to each other. And so that actually really motivated the analysis of this study, is being able to search for that.
And maybe I can jump in. This was a relatively limited data set, but I think it was a great question, is could you see convergent emergence of individual CDR3s, which you clearly see based on MHC restriction, in people responding to flu or some of the other antigens that we would see with microbial response. And so now that we have data, not only from this set, but from another rich data set and then some additional ones that iRepertoire has accumulated over the years from other investigators, I think we can look critically at how many common CDR3s there are, particularly with an effective therapy. And there's one other issue. The founder of iRepertoire, Jin Han, mentions that there's a very tiny amount of common CDR3s that we all have. And when I say tiny, it's somewhere down 0.01 to 0.1%. And I tease him because he calls that the essential index, saying there's got to be a good reason why everybody has those individual CDR3s, even if they represent a very small fraction of our repertoire. And I say, I do think that it would be worthwhile for Jen and her colleagues to look not only at convergent emergence of individual CDR3s that might be associated with kidney cancer, but also to see if there is a loss or gain of these very common, although low frequency relative to all the other chains, all the other repertoire that perhaps are either lost or gained. So we're now going to go into a deep dive to examine those elements as well.
And the other, did you look at correlation between repertoire modeling and cytokine release syndrome?
Actually, no. We don't. I don't think we have the.
Yeah, we do have data on cytokines and chemokines.
Like
a 64-plex on all of the patients. But I think, and we also have full phenotypic data. And so there's a richness there that would allow us to go back and say, all right, now that Jen and her colleagues have discerned patterns associated with response, are any of the cytokine, chemokine, and phenotypic changes that we see in the peripheral blood associated with the repertoire? And one of the challenges that I've asked her to do is to integrate these kinds of data, each of which are admittedly complex in and of themselves, and see how they cross-correlate for emergence and identification of novel biomarkers. And I think that's her next challenge.
You can always reach out, anybody with additional questions, at info@iRepertoire. Thank you so much, and be sure to stay tuned for the next iRepertoire webinar. Take care, everyone.